Good morning, guys. Can you hear me?
Yes.
Good, thank you. So, I trust your weekend was good. It's Monday now, Monday, October 19th. So, we are discussing chapter 7. We still have chapter 8 before the next test, right? Chapter 8 deals with the DNA and nucleic acids. So, today we're going to be finishing sugars, carbohydrates. So, specifically, we'll talk about polysaccharides. So hopefully you practiced some of the nomenclature and tricks we used in the previous lecture. Hopefully you spent some time this weekend doing that. And today's lecture will be mostly descriptive. So we'll talk about polysaccharides and not so much about how they're linked and so on and so forth. So we're not going to actually discuss the nomenclature. We already actually going to use the nomenclature we learned in the lecture on Friday and last Wednesday to do so. All right. If you have any questions, as usual, you are welcome to interrupt me and ask. Okay. Let's make this interactive. All right. So I'm going to share my screen. I'll share my screen. Polysaccharides. All right. So let me get a pen. All right. So polysaccharides can be homopolysaccharides when you have just one monomeric unit. So remember what poly means. Poly means multiple, right? So like polymer. Polymer, you have monomer and polymer. So polysaccharides are those that have multiple units, and you can have homo or hetero, right? One thing that actually is important, if you do ever write saccharide, there's a very interesting spelling here. There are two C's as opposed to one. Something that I learned a lot when I actually was writing my PhD thesis, because I had to use saccharides in my PhD thesis, and so I had to correct my spellings in several places. So, all right, so you can have linear, right? One type of glycosidic bond. Remember, glycosidic bond is the bond that connects an anomeric position of one chain with the hydroxyl group of, could be the same chain or a different chain, but it's going to be a glycosidic bond that forms a ketal or acetal. Or you can have branched. So branched, so here you have multiple types of glycosidic bond, right? So the anomeric position can be linked to hydroxy group at different positions on a different sugar. Now, they do not have, polysaccharides do not have a defined molecular weight, right? So these are not made by templates, right? With proteins, we have a template, right? We have a ribosomes, we have a messenger RNA that comes to ribosomes and serves as a template to read the amino acid sequence. So here we don't have a template, and so polysaccharides in general do not have a defined molecular weight. You can only talk about average molecular weight, right? All right, so they're in general, they're in state of flux. So remember I told you that acetals and ketals, and specifically hemiacetals and ketals, their formation is reversible and their decomposition is reversible. And so polysaccharides are generally not fixed molecules. They generally in a constant state of flux, right? So monomer units are added and removed as needed by the organism. So one example we looked at, I told you, for example, glycogen, right, in the liver or some in the muscle cells that is required for production of glucose as a fuel for brain, right? Constantly in a state of flux. You eat, you get some more glucose from your food, it's converted to glycogen in your liver, and then as your brain is starting to listen to me in the morning at 9 o'clock, your glycogen is starting to, as your glucose depletes in the blood, your glycogen is starting to decompose in the liver, and more glucose is released into the blood from the liver to go to the brain. So constant state of flux. And that's possible, remember, because of the chemistry of the acetal and hemiacetal functionality. So I told you that those reactions are reversible. So here are homopolysaccharides, right? So the same type of sugar, same color. Now these are heteropolysaccharides. You can see different types of sugars. This is unbranched, right? One type of glycosidic bond. Now you can see here, there is one glycosidic bond and there's another glycosidic bond at a different position. So, so this will be branched, and we will see examples of both. And so here, unbranched heteropolysaccharide and branched heteropolysaccharide. All right, so for glucose, so most homopolymers we're gonna deal with are those of glucose. So glycogen we just talked about. So remember alpha 1 to 4 linked chains. Let's see. This is something we're going to look at that in a moment with the starch, but it has the same type of linkage. But you should be able to draw this already now, right? So this means, what does this mean? This means position 1. Remember, position 1 is the anomeric position, right? Alpha, that means opposite to the C6 carbon, CH2OH. You remember that, right? And 4, that's going to be linked and formed. It's going to form an acetal with position 4 of the next glucose unit in the chain. And with glycogen, now there are branched points. Now, what that means is that every 8 through 12 residues, every 8 to 12 residues, you will see that the anomeric position of a glucose is linked to position 6 of a different glucose molecule. Not molecule, sorry, unit. It's just all one big molecule. So, so here a branching will occur. Molecular weight reaches several millions and functions primarily as storage polysaccharide in animals. And starch will be similar only in plants, right? So here's starch, also homopolymer. So now here, this one is a mixture of amylose and amylopectin. So amylose is just unbranched polymer of alpha 1 through 4 linked residues, okay, no branching. And amylopectin is going to be a different molecule, right? So this is one molecule, one large molecule, and this is a different molecule. So it's a branched like glycogen, but the branching occurs, again, alpha 1 to 6 occurs every 24 to 30 residues. So with amylopectin, the branching is not as prevalent as it is with glycogen. So molecular weight of amylopectin up to 200 million. Storage polysaccharide in plants. Okay, so let me ask you something. I don't know if you've thought about these storage polysaccharides. Let's look at the figure and then at these structures, and then I'll ask my question. So this is amylose in starch, right? So here's the glucose unit. Here is the alpha glycosidic linkage, right? You see it's alpha because it's opposite to CH2OH, right? And it's linked to position 4 of the next. So this is going to be non-reducing end. This is a reducing end. So non-reducing end of next molecule of glucose. Alpha 4, and so on and so forth. So this is a reducing end, that's non-reducing end. And amylopectin is shown here, right? So this will be, again, you have this linear polymer, but at some point, so every 24 to 30 residues, there'll be branching, where the anomeric position of the branch will actually be linked to position 6 of glucose, and you start a new branch, right? All right, so the question is, why do we need to store glucose as molecules of glycogen or starch? Why can't you just think of, let's say, like some kind of vesicles? Let's say you have some kind of, so liver cells, for example, are adipocytes, right? Sorry, hepatocytes. Hepatocytes, let's say you have some kind of vesicles that can store glucose in them. And then when you need that, it will be released. Like we talked about with neurotransmitters, right? Neurotransmitters are stored like acetylcholine or serotonin or dopamine, right? So those are stored in presynaptic cells, neurons, as in vesicles. And when the action potential arrives, they're released into synaptic cleft. So why not the same thing happens with glucose?
Doesn't it require more energy to get them out of the vesicle instead of just converting it from glycogen?
You think so? I think it's more energy because you think about you making large molecules. I mean, now there are enzymes. Obviously, there's glycogen. There's glycogen. What is it?
Synthase.
Synthase is a glycogen synthase.
Yeah.
There's glycogen synthase kinase GSK. So kinase activates that. So glycogen synthase will make that. So you have to commit a lot of resources to make glycogen, right? And to break it down.
Does it make it more available if it's out of the vesicle?
So why is it more available? I mean, you have to make this large molecule, then you have to break it down. So it looks like you have to make and break a lot of covalent bonds to store glucose, as opposed to just simply storing glucose molecules by themselves.
Is it
to store the glucose molecules when you have to shuttle them to and from?
Say it again. Sorry, start again because you were saying two of you were speaking at the same time. Say it again.
Isn't glucose a very active reactive molecule?
It's reactive? Not really. I mean, it's a hemiacetal. It doesn't really have reactive groups. I mean, it can polymerize to form glycogen, yes. So there's something about storing large number of molecules in the same place that is not biologically favorable. Just think about some kind of property related to concentration.
Would it be because they're too large to go along the concentration gradient of a cell membrane?
So there's something about concentration gradient that's good. So concentration. Is large concentration a good thing? I mean, how can you handle large concentration of something? Not easily, right? Just think about osmosis. So I think the key word here is osmosis. That's what I was kind of expecting from you. So if you have, remember, osmosis is the property that does not depend on the size or molecular weight of molecules. It depends on their number, right? So now imagine, let's say you have 100,000 molecules of glucose, right, in one molecule, as opposed to 100,000, sorry, in glycogen, let's say you have 100,000 units of glucose, as opposed to 100,000 molecules of glucose by themselves. Now imagine how much osmotic pressure that will create. Whereas with glycogen, it's just one molecule, right? So, I mean, that's the amazing thing about the osmosis. It doesn't depend on the size of the molecules, just their numbers, right? And so that's why glucose is polymerized into glycogen or starch to reduce the osmotic pressure. And you can store huge amount of glucose units that way. All right, anyway, that was just a primary reason behind actually storing small molecules like that. All right, so glycogen starch are insoluble due to their high molecular weight, often form granules. And so these granules contain enzymes. So obviously, all these numeric positions will attach themselves to hydroxyl groups on glucose, position 4, position 6, right? So there'll be lots of there'll be one reducing end and many non-reducing ends, right? So you start with one molecule and then you continue on non-reduce. So this is reducing. Then you continue on with non-reducing, and start branching and branching and branching. All right, so all these will not have the numeric position free. So this will be non-reducing. Okay. And so another very important thing is that now think about it. If it's only it's not that the enzymes that will break it down, they will not just start from the reducing end and start chopping the molecule or start from here. Basically, the enzymes can basically cut the molecule in many different places, right? So basically, the processing of starch and glycogen occurs simultaneously in many different non-reducing ends. All right, and the next polysaccharide, which is going to be cellulose. So cellulose is the difference between cellulose. The major difference is it's a beta glycosidic linkage. Now there is it's a very strong polymer, primarily because as we will see the beta linkage, there's something called anomeric effect. So we're not going to cover it here, but it's covered in more advanced organic chemistry classes. But in general, if you have a, let me draw something for you. So let's say, is this alpha or beta? Tell me, is this alpha sugar or beta sugar? I'm just drawing this in a chair conformation.
Beta.
The one on the left is beta.
The one on the left is alpha.
Alpha.
The one on the left is alpha. The one on the right is beta. So it turns out that alpha sugars, so this is alpha, this is beta. It turns out that when you have the alpha orientation, there's a lone pair of electrons here on the oxygen, which can be donated to kick out this oxygen, right? So it's beautifully positioned stereoelectronically. You can see basically it comes from behind, right? Remember, like SN2 reaction. So this lone pair comes from behind and breaks this carbon-oxygen bond. So this one cannot do that, right? You have carbon-hydrogen in this position. So so alpha enomer is destabilized destabilized by anomeric effect. So this is something that you can only learn in the biochemistry class taught by an organic chemist. You can ask any biochemist and they will not be able to tell you what an anomeric effect is actually. Well, they can tell lots of other things, but this is just specific for organic chemistry. Right, and so the anomeric effect destabilizes the alpha anomers. And that's why alpha, in most cases, things like glycogen and the starch, they're in constant flux. They use as storage, right? Cellulose, which is beta, which is not going to be destabilized by the anomeric effect. It's absent here, right? It's primarily used as a structural component, right? It's very strong. And it's very hard to break the beta cell, very hard to break the beta glycosidic bond. And that's why, you know, we can't really digest it because it's just so much energy investment, right? So, but as we know that there are, so it's probably the most abundant, not probably, it's the most abundant polysaccharide in nature, right? Because of its occurrence in plants. So for example, cotton, nearly pure fibrous cellulose. And the structure is such that there's a lot of hydrogen bonding, you can imagine. And the stability of the glycosidic bond, which is beta, which is not subject to the anomeric effect. So beta 1 to 4 linked glucose units. All right, so it's a water insoluble, difficult substrate to act upon. Most animals cannot use it because they lack the enzyme to hydrolyze, right? So these enzymes are going to be very costly to the organism because they're just going to have to consume a lot of resources and they have to be highly sophisticated to be able to break the beta glycosidic bond, which is not destabilized by the anomeric effect. So, but there is an enzyme, cellulase, that does that, right? And cellulase, so they produce, for example, by various microorganisms. They live symbiotically, right? With cattle, for example, with ruminants, and also termites. How do termites destroy homes? Because they produce the enzyme cellulase. They produce the enzyme cellulase. They can destroy wood, right? Can destroy wood, and if you can inhibit cellulase, then you can preserve a home. But in general, also cellulases are not just bad guys, they're actually very good guys because if you consider the alternative energy, right? One of the sources for alternative energy is biomass. So it's not all about petroleum, right? So if we think about biomass, which is renewable source, right? So petroleum is not renewable. You just dig it all out and then you're done. And there's no more left for our kids or grandkids. But if you can learn how to convert biomass into energy, and one of the enzymes that can help you do that will be cellulase, obviously. Okay, any questions? All right. So chitin, we already mentioned chitin in this class. So the only difference between cellulose and chitin is this position 2. Position 2, where we have, instead of the OH of glucose, we have N-acetyl glucosamine, N-acetylamine basically. It's an amide functionality. But the same, it's a beta glycosidic linkage, right? So the oxygen is on the same side as the CH2OH, right? So this is the anomeric position and the CH2OH on the same side. All right, so exoskeletons of insects, spiders, crabs, arthropods, crustaceans, and other creatures. That's a beetle that has a very strong chitin shell. So agar is a very funny one because it's a pretty complex structure. Occurs in seaweeds, and as you know, that agar is used in laboratory, for example, for electrophoresis, right? So in terms of agarose, which can form a gel, highly viscous material that can be used to separate large molecules. And so we will use agarose later in this class when we talk about DNA electrophoresis. And this is the structure of agarose, right? So there's some kind of, it's a bicyclic structure. You have some interest in bridges and so on and so forth. All right. So next type of sugars. Yes, I saw that you guys wanted the names of structures that I want you to know. Somebody put in the discussion. I saw that in my email. So I'll do that later, maybe today. I'll give you the structure of all carbohydrates that I want you to be familiar with. And obviously these starch, glycogen, cellulose will be on the list. And glycosaminoglycans will be on the list as well. So these ones are linear polymers of repeating disaccharide units. So the one monomer of the two is N-acetylglucosamine or galactosamine. Okay, so it's going to be one. And the other is going to be negatively charged. So basically, it's going to be uronic acid. Remember glucuronic acid. So we're going to look at the structure in a moment. And many of these carry sulfate esters. Sulfate esters. So these will be negatively charged as well. So many of these are extended, hydrated. So if there are sulfate esters, just think about it. Let's say you have sulfate SO3 minus hold on a second. SO3 minus, oops. SO3 minus, and so on and so forth. So lots of negative charges. And so what that means is that these will be hydrated. So the molecules of water will sit in between these negative charges to block the Coulomb repulsion between them, right? So remember the to block the electrostatic repulsion. Remember, water has very high dielectric constant, and remember, high dielectric constant blocks the interaction between charges. And so water, when it inserts in between these charges, it minimizes the repulsion between them. And so many of these are going to be extended, and many of these are going to be hydrated. And because they're hydrated, they form extracellular matrix and connective tissue. Connective tissue, lubrication of joints, right? So just think about connective tissue, very soft, lubrication of joints, right? So you can actually press on that. And so what happens if you, with the joint, for example, one major property of these kind of types of tissue is that if you press on them, they're not brittle. They're not brittle. They'll succumb to the pressure, and they will reduce their volume. Then you remove the pressure, and then the volume will be restored to where it came, to what it was originally. Why do you think that's happening? So just think about it. So these kind of tissues are resistant to breakage, right? And the resistant to breakage is because they allow for this pressure to squeeze them, and they can reduce their volume without breaking. And so the main reason for that is these molecules of water, right? So if you squeeze a molecule like that, what will happen? The water will come out. The water will come out. The negative charges will come closer, right? So they will resist any further pressure, but the whole thing will not break. But then you release the pressure, the water will come back in. The water will come back in, the charges will be separated, and you will restore the original volume of connective tissue. Does this make sense? So water basically coming in and out, and what the attractive force for the water to come in is the separation of charges, right? And okay, does this make sense? I hope it does, because I think it's a very beautiful mechanism on how we can regulate and stay and keep our important muscular skeletal system intact and not let it break easily. All right, so so these kind of glycosaminoglycans, one is hyaluronate. For example, actually you can buy this at the pharmacy, right? If you have issues with joints. Arthritis, for example. Remember arthritis. What does arthritis do to your joints? What is arthritis?
Inflammation of the joints.
Yeah, so what is arthritis? What kind of disease is that? It's a horrible disease. But what specifically causes that?
It's a degenerative disease.
Yeah, so the joints have been destroyed by what?
I believe it's the lack of like synovial fluid in between each joint.
Isn't it by the immune system?
Yeah, I think that's what I was looking for. So the immune system. Basically, if you have an overactive immune system, right, it's out of control, then the immune cells will think that will basically take these tissues, will think that these tissues are foreign, and they specifically will destroy them, right? And so for example, people with arthritis, they take immunosuppressants. And so, for example, nowadays with the COVID, people who have arthritis really having a hard time, because you don't want to take an immunosuppressant when there is COVID infection going on. So, but that's the whole point, right? So if you have a joint disease, then hyaluronate is something that can actually replace the destroyed tissue. So you can buy this at the pharmacy. Chondroitin sulfate as well, right? So you can see, so the structures of these glycosaminoglycans. So disaccharides, right? Disaccharides. So one of them is going to be the acetylated amino function, like as acetamino function instead of the hydroxy group here. And the other is going to be glucuronic acid. Glucuronic acid. So hyaluronate does not have any sulfates, but chondroitins 4-sulfate does have a sulfur. This one only has this negative charge. This one has two negative charges, one from the glucose and one from the sulfate. Now this one is keratan sulfate. Now heparin you can see has a lot of negative charges. Anybody knows what heparin is used in medicine for? Heparin is a drug, right? It's again, it's a polysaccharide, but it's a drug. It's injected into the bloodstream, when somebody has an possibility for blood clot formation, or somebody has a blood clot already formed, right? So basically it dissolves blood clots, and it is often administered to patients before surgery, right? Because surgery is always accompanied by a risk of blood clot formation. Obviously, it's an open wound, right? It's an open wound, and so there's a possibility for blood clots that can detach from the wound and travel and block arteries somewhere. So heparin will dissolve those. And how does heparin work? Okay, so prevents blood clotting by activating protease inhibitor antithrombin. So let me look at that in a moment. You can look at this slide a bit later. There's very little information on this, but this is how heparin works. So there is a protease known as thrombin. Thrombin is an important protease in the blood clot formation, and there's a natural inhibitor for this known as antithrombin. But the interaction between thrombin and antithrombin is very poor. So the affinity is very poor. But heparin comes in, and you can see here, now whenever you see a molecular model in representation, like for example, a protein shown on a screen, always look for the blue and red regions. So blue regions, that means positively charged regions. Red regions means negatively charged regions on the protein. So blue, positive. Red, negative. So what that means is that when you see blue on a protein, that means it has a lot of lysines. What else does it have?
Arginines, histidines.
Arginines, histidines, good. So blue will have nitrogens that will be protonated and positively charged. Now, when you have red, then what kind of amino acids do you expect there?
Aspartate, glutamate.
Aspartate.
Yes. So you can see here with heparin, which has all these sulfate residues. Right. Go back to the structure. You can see this heparin 1, 2, 3, 4, and there's a fifth negative charge. So disaccharide will have 5 negative charges. So all these sulfates will very nicely form the electrostatic interactions with the amino acid residues on thrombin and amino acid residues on antithrombin, and bring these two together. When these are linked, then antithrombin will block thrombin, and thrombin is one of the key proteases in the blood clotting cascade. There's actually a huge cascade, which involves numerous proteases, each cutting up some other protein and so on and so forth. And so if you take thrombin out, then the blood clot formation is actually decreased quite significantly. All right. So you can look at this table. This is basically the saccharides we've talked about, right? Look at this table. I'll just briefly go over some main points of other types of glycoconjugates. So, and assign you some pages to read, so that you can get some more information. But again, this is so descriptive. There is not really no more nomenclature for you to figure out or draw any structures, just to remember things. More like biology rather than actually chemistry. So glycoproteins are proteins which have carbohydrates attached to them. And as I mentioned to you, actually half of mammalian proteins are glycoproteins, right? So whenever we talk about proteins, always keep in mind that these proteins have sugars attached to them. And how they attach to them, there are two primary sources of attaching a sugar to protein. Is one through a serine residue on a protein, right? So this you can see serine, oxygen, attached to an anomeric position of galactosamine here, N-acetylgalactosamine. And here is asparagine, nitrogen of the asparagine is attached to through the numeric position to N-acetylglucosamine. So it can be, so glycoproteins can be O-linked or N-linked, okay? And then you can see usually these carbohydrates, they're not long chains. They're not like glycans. They're usually pretty small collection so this is going to be reducing end, non-reducing on the left side. So small collection of carbohydrates linked through glycosidic bonds. And you can see that serine or threonine, these are all linked, and asparagine will primarily give you N-linked. And most proteins, protein-protein interactions are mediated by their carbohydrates. So when we talk about the two proteins interacting in a cell with each other, most likely that's because their carbohydrate units come into close contacts. And glycolipids. So obviously, you can also form conjugates where you have carbohydrates attached to lipids, and this is, for example, a structure of a lipopolysaccharide. Lipopolysaccharide, remember, on gram-negative bacteria, there is a lipopolysaccharide coat, which protects the peptidoglycan layer underneath it, protects the cell wall. And this is the general structure. So lipid A is a pretty conservative portion of it that doesn't change from one lipopolysaccharide to another. Now there's some more detailed structure. This is the core that doesn't change very much. And now there's an O-specific chain. O-specific chain is a bit longer, and so on and so forth. So again, lipopolysaccharide in many cases it's used for as a barrier, for example, to stop penetration of small molecules, the toxins. In other cases, lipopolysaccharides are important for cell recognition, right? So, for example, how do bacteria infect other species, other cells, other organisms, is through this kind of recognition. And remember, carbohydrates are the key molecules in biology for recognition, right? All right. What else did I want to mention? We have a few more minutes. Proteoglycans. So don't confuse these with glycoproteins, even though they sound similar, versus glycoproteins. So glycoproteins, remember, are primarily proteins, which have small carbohydrates attached to them. Proteoglycans, on the other hand, have long carbohydrate chains attached to proteins. Now there is syndicans and there is glypicans. So let's look at both. So these proteoglycans will have the following structures, two types. So for syndicans, you have a protein that crosses the cell membrane once, and then there is a glycosaminoglycan attached to the protein. So we talked about heparan sulfate, we talked about chondroitin sulfate, right? So this is going to be glycosaminoglycan attached to the core protein, which is going to be syndican. Now in this case, we have, so it's a glypican, which is, now it's a protein which does not cross the cell membrane, but it's attached to cell membrane through something which is called GPI anchor. Glycosyl phosphatidyl inositol anchor, GPI anchor. So there's a lot of research actually into GPI anchors. We'll talk more about GPI anchors when we talk about lipids. But they have this general structure. They have some fatty acids which serve as an anchor, right? This is how this whole thing is actually anchored to the cell membrane through these fatty acids. And then there is a carbohydrate unit, which can also involve a cleavage site. And then there's a protein. There's some disulfide linkages, and also there is a glycosamine and glycan chains attached to that. Now, again, in most cases, so these form the what's known as extracellular components of extracellular matrix, ECM. Whenever you see abbreviation ECM, you should recognize this as extracellular matrix, right? So there are cells, and the cells bathe in the extracellular matrix. And so these sugars form one of the main components of extracellular matrix, right? So these cells basically find themselves in extracellular matrix. So for example, if you think about, let's say, cancer cell wants to metastasize, right? It has to find its way through this extracellular matrix, right? So let's say this is a cancer cell and it wants to get through. And so this cancer cell has to release enzymes which will break down the extracellular matrix, right? So the cancer cell can escape its original location and travel to the bloodstream, get into the bloodstream, and be carried to remote sites in the body. All right, so these are glycosamine. Now this is just a more detailed structure of one of these proteoglycans. So this is a glycan unit. You can see there is just there is some glucose, N-acetyl glucosamines. There's glucuronic acids. There's some sulfates present in here as well. 2-sulfate, 4-sulfate, iduronic acid. This is a different type of sugar. And you can see that actually, basically, there are alternating sulfated domains and non-sulfated domains. I don't know what A stands for, but this is sulfated and this is non-sulfated, right? So just a more detailed structure of these proteoglycans. So different glycosamine and glycans linked to the core protein. Linkage for the, okay, how are these linked? Let's look. So this will be our core protein, right? Let's say this is syndecan, which crosses the cell membrane. On the outside, it will have this serine. Now, in general, these are pretty similar. So there'll be serine, then there'll be glycine, there'll be something else, could be anything, and there'll be glycine as well. So whenever there is a combination of these 4 amino acids, then most likely the serine, which starts this combination, will be linked to glycosamine and glycan. And usually this starts with these 4 sugars. This is xylose, galactose, and glucuronic acid. Glucuronic acid, these are anomeric linkages, right? Anomeric linkages for you. And this will be chondroitin sulfate. Chondroitin sulfate is again the repeating unit of these disaccharides, which are sulfated. All right, so there are a couple of slides that actually I'll just ask you to read. There's not much here. Just more about how the joints work, and specifically about proteoglycan aggregates, which are separate molecules, and again provide lubrication because they hold a lot of water. So low friction material, so primarily form cartilage. Okay, I'll just assign a reading for you. Let me actually maybe give it to you right now. So proteoglycan aggregates, that page, let me go to the end. Read, oops. Read pages 264. .266, so just 2 pages of slides that we didn't have time for. All right, any questions? All right, so we're done with chapter 7. Homework is due Wednesday, do not forget, okay? And do homework, do not leave it until Tuesday late at night, right? And more discussion, please, more discussion, and let me know if you have any questions. And I will talk to you on Wednesday.
Thank you.
Yeah, bye guys.